So I'm going to talk about individualized patient care through precision medicine. So I'm going to combine a number of concepts. The concept of personalized medicine that we're all talking about, that makes sense because we know better about the pathophysiology of several diseases. We're able to target certain genes or manipulate certain proteins, and we use precision medicine for that. But what I'm going to, my thesis today is that even in an area that is really exploding, which is rare and congenital diseases, where definitely we know already the gene that we are targeting, we think sometimes that because we know how to deal with these kind of diseases, we don't need to do so much in terms of personalizing both the selection of patients and the way we monitor them. That's where I'm going to demonstrate to you how important it is to make a shift maybe in that direction. And you mentioned my previous experience with companies in the diagnostics industry, like Abbott Labs. That's where I started my career. I also work with Becton Dickinson. I've been working as chief medical officer with some diagnostics companies. But in the past few years, and also in my previous experience in the clinical development of biopharmaceuticals, I started, by the way, with the Humira team, as you mentioned, and I was working with the rheumatology type of franchise there. And in my past 10 years working with gene and cell therapies, I've been trying to incorporate all these ideas. So what I'm going to be discussing with you first is in the concept of individualized medicine in patient care. I'm going to take you step by step to ensure that we understand the relevance of biomarkers for patient selection and biomarkers for patient monitoring. And then I'm going to use my two case studies right now with ASC Therapeutics. We have a gene therapy where we're looking into treating patients with Hemophilia A. We're using both traditional gene therapy and also CRISPR therapies, and also stem cell therapies. And we are treating graft versus host disease, which is a very serious condition that happens in patients that receive bone marrow transplantation. So starting with the way we see today personalized medicine, which is mostly genetics driven. So we have the traditional one size fits all. And again, one very good example is Humira, where we practically, once we define a disease target, and I was involved in several clinical trials for rheumatoid arthritis especially, and also for other rheumatic diseases, we knew that there were a number of patients that were not responding. And but still, the agency, the FDA, approved the drug because it was significantly higher the results versus placebo. That's clear. That's the way all these drugs are approved. Now, there are a number of patients that are not responding to Humira, to anything, besides being Humira the best-selling drug in history, by the way, and it still is at the top. Now, when we look into personalized medicine approach, now we know the genetic drivers, genetic profile that tells us these are the patients that should receive this therapy. And then others which do not fit with that profile would get the standard therapy. Now, this is not sufficient. This is not just about genes. There are also several aspects in your family health history. There are several other factors that influence and impact the risk of the disease that you have right now, or I mean you in general, and the future medical concerns. There is diet, environmental factors, age, body mass, weight. So there are a number of aspects that need to be taken into account when considering on having this personalized treatment. And when moving now into biomarkers, now we see that this is the definition by the FDA. What they're saying is that these are biomarkers that provide information essential for the safe and effective use of a corresponding therapeutic product. That's what the FDA says. But at the end of the day, when we present a new molecular entity or a new drug to the agency, what they're focused on is on clinical efficacy and safety. And they tell us, okay, sometimes if you have what they call a surrogate endpoint, it's also going to be acceptable. And that's where we have all these omics, which are genomics, transcriptomics, epigenomics, proteomics, phosphoproteomics, metaboproteomics. All this is absolutely essential because some of them are going to be useful for certain type of clinical trials, others are not. At the end of the day, we take these single biomarkers, we combine them with AI or with machine learning, and we create signatures. These signatures are telling us which patients should be included in our treatments and which ones should be excluded. So all this is becoming reality, but this is a process. And this is what I'm trying to communicate, that we still are not there. We have been talking about combining diagnostics with therapies. The agency has even created an area or a department that is working so-called companion diagnostics, but we are still, believe it or not, in the early stage. Now, when we talk about biomarkers, we have to see them from two perspectives. First are the ones, and again, I'm giving also you the perspective from the agency, from the FDA and other regulatory agencies. First, we need to make sure that we select patients, and now I'm starting to zero in gene and cell therapy that we're going to be talking about in a moment. And this is first in terms of genotyping of the target genes or phenotyping of the target cells, or obviously the proteins that we are evaluating when it's other areas. Now, in many cases, we may have also the opportunity to understand how that drug works, working ex vivo on certain target systems or organs. And the most standard one is kind of immune response systems. And once we have identified those patients, and that's also where sometimes I think that the industry is not focusing so much, is being able to monitor the patient response. We're talking here about therapies that are extraordinarily expensive. So we have Zolgensma recently approved, recently, a few years ago, and patients, one single infusion is about $2 million. These patients obviously are followed up very thoroughly, but in many other cases, I see clinical trials in other diseases where it's not so clear that patients are being followed up so thoroughly. We're going to incorporate a number of biomarkers in our studies, which are going beyond what other companies are doing in this space. So it's important to ensure that we identify efficacy biomarkers related to the gene or cell product clinical activity, and also safety biomarkers related to expected adverse events. So it's not just the clinical aspects, the clinical manifestations. It's also taking a good look into these surrogate biomarkers. Now, believe it or not, everything I've been talking about is personalized medicine. And what that means, and you see this example on clothing, is when you have certain clothes that are ready to wear, they're off the shelf. You give the right drug to the right patient when you identify a number of biomarkers or indicators or demographics or a number of aspects that tell you this is the right drug to the patient. Where we're moving towards, and that's what the message I'm bringing today, is individualized medicine, where we are even creating the right drug for the right patient. And this is something that is starting to take shape. And it's going to be very difficult, obviously, to have one company developing these kind of made-to-measure type of drugs. But what we're going to have is different drugs that are going to be very similar, probably from even different companies, that are going to be given to the right individual. And that's what you're going to hear in a moment. That's what we are doing at ASC Therapeutics. We're creating one of these drugs that is going to be relatively similar to others that are going to be in the market in terms of gene therapies, but still with certain characteristics that differentiate that. And the advantage that we have, and everyone working in this space, is that the FDA understands this, and they are fully committed to help us to help the sponsors move forward and ensure that we overcome limitations in manufacturing, that we really individualize the product development, that we provide input and collaborate in terms of novel endpoints, and they are encouraging even clinical trial, new clinical trial designs. So there's a lot going on. And I can tell you this is all happening in real time and at a very, very fast pace. So now let's move into my case study. So this is a way to show you and to practically put into a real life example what I've been talking about. First of all, I would like to say that what is happening right now in the biopharmaceutical industry is absolutely revolutionary. I don't think we have ever seen this explosion of knowledge and know-how going on, and in terms of not only from my perspective in clinical development, but also in manufacturing, in R&D, in clinical pharmacology. So all this is taking us, and just if you think in the middle, this large biologic, that's what I was working on in early 2000s with Humira. It was a biologic. It is a biologic, which is a very complex type of molecule, which requires bioreactors, requires also a number of steps that make this biologic really almost a living type of, obviously, a monoclonal antibody. Now we moved into pegylating them, making them bigger molecules. But when we talk now about gene therapy, I don't think I can compare that to anything else than moving from a jet fighter to the International Space Station. It's absolutely mind-blowing, everything that comes into being able to develop this final vector that is going to incorporate the information on that you are going to deliver to the patient, and it has to be really targeted to the patient and to certain cells. And by the way, there is an accelerator on all this happening right now, and this is the mRNA vaccines for COVID, which really are helping us, believe it or not, the gene therapy companies ensure that we have a more streamlined type of manufacturing. We're learning, and this is really spectacular what is happening. So I'm going to talk about hemophilia A. This is a condition where you have patients that are missing one of the key factors in the cascade, that really in the coagulation cascade. Patients that really miss this factor, either factor A, which is leading to factor 8, that is leading to hemophilia A, or factor 9, that is leading to hemophilia B. There are a number of patients out there. They are getting right now replacement therapies, or some therapies that are called bridge therapies, that practically bridge the gap in some factors. But there are still a number of patients, for instance, that cannot even be treated with gene therapy because they have inhibitory antibodies that inhibit the viral vector that we're using, or sometimes there are even patients that cannot even receive any type of replacement therapy because they have antibodies against factor 8. And those patients receive different factor 8s from different individuals, from different species. Like, for instance, there is something called Obisur, which is fully developed from porcine sequences. So it's a factor 8 from the pig, basically, but is given to patients with acquired hemophilia A. So there are different ways to individualize and use different products depending on the patient. Now, in our case, what the industry is doing to treat these patients with hemophilia A is ensure first that you identify a target. And the target in this case is the hepatocyte. This is a critical liver cell that definitely is producing hundreds and hundreds of proteins, but is not used to be used as a biofactory the way we are doing that. So we are forcing the hepatocyte to take the virus, to incorporate in the nucleus the transgene, and you're going to see how all this works, and being able to produce factor 8. But the hepatocyte doesn't produce factor 8 regularly. It's another sinusoidal cells in the liver. So we're forcing the hepatocyte. And the way we do that, and I'm going to use this analogy here from that I call from the lab to the hepatocyte. And this is an analogy from the Apollo 11 historic journey to the moon. And you're going to see that at the end of the day, we have a rocket, we have a propulsion capsule, we have a lunar module, and we either land in the moon or we are orbiting the moon. And you're going to see how all this works. First of all, we have a viral vector, which is called AAV8, is a harmless adeno-associated virus. We use 8, others use 9, 5, there are different type of viruses. And it has a liver-specific promoter. A promoter is a kind of capsule that has all the elements to ensure that it can release the transgene, which is the factor VIII transgene that we have created in this construct. We call it a construct. So it includes the actual factor VIII, all the genetic information from the factor VIII, which in our case, and this is where I come to the individualized aspect of what I was talking about, we have incorporated 9% of sequences from the porcine factor VIII. Why do we do that? Because we license this entire construct from the Emory University in Atlanta, because they had been working for over 20 years in phylogenetic aspects of factor VIII. They were the ones that developed this 100% porcine factor VIII use in certain patients, and they identified exactly the sequences that were going to increase the secretion and also the biosynthesis of factor VIII. So that's what we have. We also, in order to pack this factor VIII into the rocket, into the virus, we need to cut it. We need to take certain portions of it out. It's called the B domain deleted. So we have A and C, but not B. And this is something that the entire industry does. So this is our individualized product. This is a product that we are going to start treating patients in the next few months. I hope to have an IND filing in the next few weeks. So we had already a number of preclinical data that informed the phase 1 clinical trials. We saw that the protein levels were actually elevated in animal models, including monkeys that received this construct. We saw some inhibitors in some animals, but they are not going to happen in humans because they're just for monkeys that don't recognize the human factor VIII, and we didn't find any adverse events. So besides using this traditional approach, where what we are doing is that we're incorporating, and this is what happened with what I described to you, we were incorporating this transgene into the nucleus, and it was orbiting the chromosomes. So it's not incorporated in the chromosomes. That's the way everyone in the industry is working right now for gene therapy. You are, the moment that you have any kind of cell that divides, you lose this transgene because you treat these patients only once. This is an infusion that happens only one time. So to be able to treat children, which, as you know, in these genetic diseases, and one example is Hemophilia A, you need to also make sure that you are able to maintain the production of factor VIII indefinitely, even if this liver cell is dividing. So for that, we are using CRISPR gene editing. I'm sure you know CRISPR. Last year, it was the Nobel Prize from our neighbors up here in Berkeley University, and also in the East Coast. What we do here is that instead of giving this promoter with the learner module, which means the way to activate our transgene, we just incorporate the transgene in a certain position in the chromosome, and we do that very close to the albumin locus. The albumin is one of the most ubiquitous proteins in the body, and it has a very strong promoter. So we put that close to that between exon 13 and 14. We did all the work on that. Our company has been working in this space over 12 years in CRISPR and so on. So when we do that, the plan is to be able to treat not only adults, but also children. So we're going to incorporate this transgene very close to this locus. Is going to be in the hepatocyte, and we are going to have a lifetime effect. The corrective factor VIII transgene is going to be now established, integrated, and can be used for patients. It has a high potency and efficacy, and is safe. So this is everything that we are doing. So we have these two products. They're going to be individualized to the type of patients that we think are going to require that, depending on their needs. So in general, with those two types of therapies, gene therapy and CRISPR, gene editing clinical trials, the idea is to ensure that we first identify patients with very severe hemophilia A. Then we have a number of selection biomarkers that are going to tell us if these individuals already have inhibitory antibodies to the factor VIII protein, to the rocket that we're using, which is the adenovirus 8. And also we're going to do a number of liver function tests and so on. So very important here, and as chief medical officer, I'm also very careful that these patients have a pristine liver, I call it, which means that these are patients that don't drink, don't take fast foods, that they are sure to understand that we're using their liver to do a very important job. So these patients for life will have to be extremely careful with what they eat and what they drink, believe it or not. And this is also a way to individualize the therapy. If you have someone that is not able to do that, we look for fibrosis, we look for a number of markers of liver function or dysfunction. And then a number of monitoring biomarkers. Once we treat these patients in a single infusion, once in a lifetime, then we follow up these patients and ensure that we understand how the immune system is responding to this new factor VIII and the potential of factor VIII inhibitor levels that we don't think are going to take place. And also obviously to see the actual factor VIII activity. So with that, I'm going to end up with another example, and I'm conscious that this is a lot of information, especially for those of you that are not in the field. For me, it's absolutely fascinating. I really enjoyed every minute in my job, and as an immunologist, I am also looking forward to another program that we have on cell therapy. And this is related to what I call the power of stem cells. And this is the type of product that we have. I'm going to explain it very briefly. To the left, you have these cells that are the DSCs, the Sigilstromal cells. These are cells that are in the placenta. The placenta is the organ, is basically a new organ that a pregnant woman develops to protect the fetus. Why? Because the fetus contains all the antigens from the father. So it could be recognized by the immune system of the mother, and it could obviously destroy the fetus. And this is something that the mammals have developed over time, so that they have a number of cells, by the way. That's what a group in Karolinska discovered, and we licensed that from them. They were working over 20 years on patient on this type of cells, on mesenchymal stromal cells, and then these serial stromal cells. And they realized that these cells had two main characteristics. They were able to increase PD-L1, PD-L2, which you want to decrease, by the way, in cancer, but it's very important here to ensure that we that these cells do that, because they are also going to identify and select the cells that can get close to the fetus, and they also increase the regulatory T cells. So that means that they regulate, they have an immunoregulatory effect on cells that could eventually attack the fetus. And on the other hand, they control any kind of acute inflammatory conditions. They induce apoptosis in those cells that could be alloreactive versus certain antigens. And when we started using these cells in graft versus host disease, graft versus host disease is what happens when you get a bone marrow transplantation. So patients that have leukemias and lymphomas and other conditions receive practically their condition. Their immune system is wiped out. They receive the cell from another individual, and what happens is the same situation as in the mothers and the mother and the fetus. You have these cells that are starting to attack organs in the recipient. Obviously, the reason you do that is because you want to destroy the malignant cells. You want to destroy the leukemic and the lymphoma cells. But they also start attacking especially the skin, the gut, and other areas. So when our collaborators in Karolinska, Sweden, started working on this now over 10 years ago with these DSCs, they started to see how they were able to control these effects, and the cells from the donor were immunoregulated. extraordinary results, and we are now moving forward into studies. And here is the same. So we also need to understand now the first, what type of patients are we treating? These are steroid refractory acute graft-versus-host disease. These are a very acute condition. Practically within a week, these patients can even die. But the moment that we start giving them the DSCs, the Sigilstromal cells, they start to improve and they start to control the entire immune response. So we need a number of selection biomarkers here. We need to identify those patients that have probably a certain immune profile that are going to respond to that. We're working on that. So that's my point. We are also looking into tissue resident immune cells in the gut and skin, which are the major target organs. And then we also look into ex vivo examples on how to understand the way what we call ex vivo functional effects on target systems and organs. And finally, once we treat these patients, just within 1 month, 2 months, we need to understand a number of aspects on safety and efficacy. We're using some biomarkers that having developed, one of them Mount Sinai is called Magic Biomarkers, and we're going to work with them, with this group in the East Coast on analyzing our patients. So again, with all of that, I just want to give you a final point on the future drivers of personalized medicine and patient care. I think it's very clear that we're moving very fast because we are already understanding the biologic underpinnings and genetic drivers of disease. All this work, the fact that we're using the biomarkers, that we are evaluating these patients thoroughly, are going to improve the way we understand these biologic changes. The absolute explosion right now on high tech and on next generation sequencing and computational biology is also helping us to advance the discovery of disease mechanisms. And definitely we're getting a ton of data, what is called big data. And with all of that, we're going to have predictive models of gene expression regulation. We're going to be able to understand the entire picture. So what I can tell you now is that what is going to happen in the next few years, what is already happening, is absolutely fascinating, and is really revolutionary. And I'm really happy to be part of it and take any questions that you can have. I think again, we are in a very good place to conquer another world, as we did many years ago with the moon. Thank
you. Okay. Thank you very much, Dr. Segurado. Very nice talk, and thank you for leaving some time here at the end for Q&A. And while people are perhaps writing in their chats, if you have a question, by all means, use the chat room if indeed you have a question, and I can try to get to your question as best I can. I'm going to start though, just out of curiosity. I'm an ex-FDA medical regulatory officer in the osteoarthritis division. Back in the old days, and I think even now, the classic small molecule play in big pivotal trials was 2 randomized controlled trials, 2 of them, replicate evidence. Are you finding that that's the same case with what you do in gene therapies and all the type of genetic work that's happening? Is the FDA generally requiring 2 randomized controlled trials?
So they are not. In gene therapy, it's very difficult to really have patients. And by the way, this is a phenomenal question, because there is a big change, as I'm saying right now. So indeed, you are expecting to have a randomized clinical trial, and you as an ex-regulator, you understand that. Sure. Now, we cannot really wait years and years to see how patients are doing without certain therapy. So I was involved with Avexis, which was acquired by Novartis, on Zolgensma, which was used for patients on spinal muscular atrophy with the SMN1 gene. And there was also just almost, you can call it anecdotal type of work done by Nationwide Children's Hospital, where they started seeing children that indeed were expected to die within 1 year or 1 or 2 years. And I remember that when I was involved with them, what we presented to the FDA, believe it or not, was videos of these kids, how they were walking around when they were 2 years old. And when they saw that, they said, we don't need anything else, basically. There was a study, there was a single arm, and that's exactly what we're doing now. With hemophilia studies, I am about to embark in a phase 1/2. It's a combined study, about 10 patients, dose escalation. But for the pivotal trial, me and others, Biomarkers and others, we just have a single arm where we treat one set of patients in it's about 120 patients or something like that. That's what I'm thinking right now. And there is no, it's not a randomization. There is not placebo controls. I think that, and that's why the fact that we have so many surrogate biomarkers, the fact that we are able to see this information on time and with patients that serve us as a kind of control arm. So that you have the patient before we're getting, and by the way, these patients need to be 6 months on observation, on pre-screening, and then we treat those patients. That's the way it works.
Very nice. I must confess I'm jealous that you don't need to do placebo-controlled trials. But,
I'm happy about that.
Yeah, that's the swamp that I swim in. So we actually have a nice question coming in. Actually, I think a very thoughtful question coming in from Vishal Mehta. A question to you, doctor, is how do you see the role of bioethics and particularly industry self-regulation with the proliferation of cellular and gene therapy? I assume that's what you mean by C and GT. So I use the cellular gene therapy.
Yeah, this is a very critical question, and I think let me go to the worst case scenario, which is CRISPR therapeutics therapy. So what happens with CRISPR is that you are really inserting a gene in the chromosome of an individual, and we are not there yet, even with our animal studies. We are working with the standard traditional gene therapy, which I can also address this question. But in terms of CRISPR, we have to be extraordinarily careful that we are really targeting the actual locus, the actual position in a certain chromosome, in a certain place. And I mentioned exon 12 and 13 just before albumin locus, because if you put that especially in a germ cell that is going to potentially, especially if you have a woman with in the fertility still fertile and able to have kids, that could be tremendous. So there is a lot of I would say self-awareness. I'm not sure if I can call it regulation, so that we are regulating ourselves. But I can tell you, we have many eyes looking into that. There are bioethical groups who are looking specifically into that. There are conferences. The FDA is not directly involved in that, but we are all very aware of that. And especially in my case, as a clinical developer, I am tremendously careful on checking what we call off-target insertions. So I don't want to insert a gene in the wrong place. And that also applies to genes, to standard traditional gene therapy, although that's not something that is so ubiquitous, and we know that in general there is no issues, but things can happen. So we're very conscious about this situation. You're right.
Very good. It almost seems like you're required, and you attest to informed consent on steroids, so to speak. I mean, that goes beyond informed consent for what you guys have to do. So terrific answer. I can only imagine some of these patients and the condition. SMA, as an example, very, very difficult condition for the family, for the patient, as it were. We'll give it a few more minutes here for questions. And again, if you have any other questions that you don't want to ask here, that's okay. You can get to the doctor by Shante. She can get you. I'm sure you'll share your email address if you like, if people have questions for you. If you weren't, sorry about that, but I may make you available to our colleagues in the audience. I don't see anything else coming in for you, and that's fine. So again, thank you very much for a wonderful talk on an emerging therapy, somewhat complex, but somewhat necessary, I believe, as well, as we take it to the next level. So again, golf clap for you, Doctor. Very, 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 very well done.
Thank you so much.
Yep. Thank you, Dr. Segurado.